Dear students, after studying this module, you shall be able to name and describe the parts of a neuron and explain their functions, describe the supporting cells of the central nervous system, describe the electrochemical process of nerve impulse conduction and the role of ion channels in the neurons, learn the structure of synapse and the process of interneuron communication. Introduction. This module introduces you to the structure and the types of neurons. It will also describe the intra and interneuron function and the conduction of nerve impulses in the nerve cells or neurons. Animal body is made up of trillions of cells. Different cells are specialized to perform different functions in a complex and integrated way. The cells present in the nervous system are different from those in the rest of the body with respect to their structure and functions. The cells constituting the nervous system are called neurons. The nervous system is known as the information processing system. It picks up information from the environment and helps give the appropriate response for the purpose of survival. It is the physiological substrate of the stimulus response mechanism. There are three types of neurons present in the nervous system according to the function performed by them. These neurons are sensory neurons. These are those neurons that detect changes in the external or internal environment and sends information about these changes to the central nervous system. A motor neuron is located within the central nervous system, which responds to the signals of sensory neurons. An interneuron, a neuron located entirely within the central nervous system, which analyzes small bits of information and is therefore also called relay neuron. Variety of neurons: sensory, inter or relay, and motor neurons. The figure indicates that the sensory neuron is carrying information from the painful outside stimuli to the spinal cord, where the inter-relay neuron analyzes the information, decides what to do with the painful stimuli, and responds back through motor neurons by withdrawing hand from the painful stimuli. Structure of a neuron. Most of the neurons have similar structure, which includes soma or cell body, dendrites, axon, terminal buttons. Soma is the cell body of the neuron which contains nucleus. It provides for the life processes of the cell. Its shape varies in different neurons. Dendrites are the branches coming out of soma. They are numerous in number. They receive information from the terminal buttons of another neuron. Axon is a long and slender tube covered with a myelin sheath. Axon carries the information received by dendrites through terminal buttons. The way in which the information is transmitted through axon is called the action potential or the nerve impulse. The structure of the neuron varies according to the way dendrite and axon leave the soma. The most common type of neuron is multipolar neuron, where one axon leaves from soma and many dendrites leave soma on the other side of it. Another type of neuron is the bipolar neuron, in which one axon and one dendrite leave soma in opposite directions. The third type is the unipolar neuron, in which only one strand leaves the soma and leads to axon with branches of dendrites and terminal buttons on the opposite sides. Terminal buttons are the end structure of a neuron. They perform a special function in which the information from axon travels down to the terminal buttons. They contain vesicles of neurotransmitters. These neurotransmitters are then received by the dendrites of the next neuron, and depending on the type of the neurotransmitter, action potential is either generated or not in the next neuron. With the help of an electron microscope, the internal structure of the neuron has also been studied. A typical multipolar neuron is characterized by the following organelles within its soma. Membrane. The membrane defines the boundary of the soma. It is made up of a variety of proteins. These proteins allow the transportation of selective substances inside and outside of the soma. Therefore, the membrane of the soma is called selectively permeable or semi-permeable. Cytoplasm. It is jelly-like free-floating substance which is present in the soma and contains small specialized structures or organelles providing life support materials for the cell. Mitochondria. It is an organelle, specialized structure in the soma analogous to an organ which breaks down nutrients such as glucose and provide the cell with the energy to perform its functions. Mitochondria produce a chemical called adenosine triphosphate, also known as ATP, which can be used throughout the cell, a source of energy. It was a primitive independent structure many eons ago, but later became the part of the larger cells. It has its own DNA. It is inherited from the mother only. Nucleus. Nucleus is present deep inside the soma. It contains chromosomes, and chromosomes contains deoxyribonucleic acid, also known as DNA. Chromosomes have important function. They contain the recipe for making proteins. Proteins of chromosomes are called genes, which in a way consist of recipe of making other protein. Protein performs many important functions in the neuron. If a neuron is grown in tissue culture and exposed to detergent, the lipid membrane and much of its internal substance dissolve away, leaving a matrix of insoluble strand of protein. This matrix is called cytoskeleton and gives a neuron its shape. Besides providing structure, protein acts as enzymes. They decide which molecule to join together and which to split apart. They decide what is to be done of the raw material present in the cell. Proteins are involved in the transportation of substances within the cell. Some of the material are required in terminal buttons and are made in the soma. These materials are transported through axon, and this transportation is called exoplasmic transport. This transport is accomplished by long protein strands called microtubules. Microtubules are the bundles of 13 filaments arranged around a hollow core. They act as railroad tracks, guiding the progress of substances being transported. The movement is in two directions, from soma to terminal buttons or from terminal buttons to soma, and is called anterograde or retrograde exoplasmic transport respectively. Energy for this transportation is provided by ATP. Supporting cells. In the CNS, more than half of the cells present are the supporting cells because neurons have high metabolic rate. These supporting cells supply nutrients and oxygen to the neurons to protect them from dying. There are mainly two types of supporting cells, glial cells and Schwann cells. Glial cells. Neurons are protected by glial cells from the rest of the body. Such cells surround neurons and hold them in place, control their supply of nutrients and some of the chemicals they need to exchange messages with other neurons. Insulate neurons from one another so that neural messages do not scramble. Act as housekeepers by destroying and removing the carcasses of neurons that are killed by disease and injury. There are several types of glial cells in the central nervous system. Three most important glial cells are astrocytes, oligodendrocytes and microglia. Astrocytes are also called star cells because of their shape. The arms of the cells are spreading out from all sides. These cells provide physical support to the neurons, clean the debris within the brain, control the chemical composition of fluid surrounding the neuron, and provide nourishment to neurons. The arms of the cells are wrapped around blood capillaries and some part of the neuron. These arms of the star cells receive nutrients from capillaries, store them and release them to neurons when needed. These cells also surround and isolate synapses, limiting the dispersion of neurotransmitters that are released by the terminal buttons. Astrocytes also clean the debris of dead neurons and other substances inside the central nervous system by pushing them against it and finally engulfing and digesting it. This process is called phagocytosis. Phagein, to eat, kutos means the cell. Astrocytes take place of dead neurons, thereby filling the gap. Oligodendrocytes, they provide support to axons and produce the myelin sheath. The myelin sheath is of utmost importance to neurons, which insulate one neuron from the other and thereby preventing the disorganization of electrical impulse moving through the axon. Myelin sheath does not cover the whole axon but covers it in series of segments of 1 mm long and leaves some portions uncoded of about 1 to 2 mm called nodes of Ranvier. These nodes of Ranvier increase the speed of transportation of electrical impulse through axon. Thus, the axon looks like a string of beads which are 80 times in length than its width. An oligodendrocyte produces up to 50 segments of myelin that wrap around the segments of axon, thus forming the myelin sheath. Microglia, they are the smallest glial cells. They act as phagocytes and engulf and break down the dead and dying cells. They also act as representative of the immune system in the brain, protecting the brain from invading microorganisms. They are responsible for inflammatory reaction in response to brain damage. Schwann cells, they are found in the peripheral nervous system. They act as oligodendrocytes of PNS. They form the myelin for the axon. The process is a bit different here. Here, each segment of axon consists of single Schwann cell and each Schwann cell wraps around only one axon. Thus, the entire Schwann cell surrounds the axon. Communication within a neuron. Communication within a neuron is an electrochemical process. Electrochemical process includes 4-stage events: resting potential, depolarization, the action potential, and hyperpolarization. This electrochemical process is understood in terms of ions, electrically charged particles, exchange such as sodium, potassium, chloride, and organic ions. There is a normal concentration of ions and the forces that keep them at place, which is also responsible for their movement. The fluid within the cell is called intracellular fluid and fluid outside the cell is called extracellular fluid. Ions predominantly present in the intracellular are organic and potassium, whereas ions predominantly present in the extracellular fluid are chloride and sodium. These ions are in constant force to move inside and outside of the cell. Organic ions are found only inside the cell and it majorly contributes to the negative charge inside the cell. Potassium ions are predominantly present inside the cell and two forces are acting on it: diffusion and electrostatic pressure. Because the concentration of potassium ions is low outside the cell, the force of diffusion acts on potassium ions present inside the cell to move outside. Also, because of the presence of organic ions inside the cell, potassium ions face electrostatic force to remain inside the cell, thereby balancing the movement of potassium ion, and therefore potassium ions remain inside the cell. Diffusion is defined as the movement of molecules from the region of high concentration to the region of low concentration. Electrostatic pressure is the attraction force between two particles charged with opposite signs, and the repulsive force is that between atomic particles charged with the same sign. Similarly, chloride and sodium ions are predominantly present outside the cell, and chloride ions face force of diffusion to go inside the cell where its concentration is low. It also faces electrostatic force from the sodium ions to stay outside the cell, thereby balancing the movement of the chloride ion, and so the chloride ion remains outside the cell. Sodium ions face force of diffusion to go inside the cell because its concentration is low inside, and also it faces electrostatic force to go inside the cell, which is predominantly negative. It forces sodium ions to move inside the cell. However, most of the time when neuron is at rest and not getting stimulated, the sodium ions do not move inside when they should because of the two forces. This movement is prevented by another force, the sodium-potassium pump. This pump continuously pushes the sodium ions outside the cell, and this movement is prevented at the cost of pushing two ions inside the cell per sodium ions. And this movement is possible because the cell membrane is 100 times more permeable to potassium ions than to sodium ions. Resting potential. Resting potential is the membrane potential of a neuron where it is at rest. It can be measured using microelectrodes which record the activity of an individual neuron. This microelectrode is inserted into the axon of a neuron, which is placed in a seawater solution. Another wire electrode is placed in the seawater. Both of the electrodes, wire electrode and microelectrode, are connected to a voltmeter which measures the electrical activity in the axon. As soon as this is done, it is found that the axon is negatively charged with respect to the outside, the difference in the charge being minus 70 millivolt. Thus, the inside of the membrane is minus 70 millivolt. This electrical charge is called the membrane potential. However, when the membrane potential is measured at rest, it is called the resting potential. It means that at this time, no ion exchange is taking place, and we may also say that membrane is negatively polarized. Depolarization. In this stage, the neuron is no more at rest now. Some stimulus has activated the neuron and has successfully caused the membrane's potential to increase the threshold potential of neuron membrane to minus 50 millivolt. As a result, the polarized neuron is now becoming depolarized. Action potential. As soon as the threshold potential of a neuron is achieved, the sodium channels in the membrane open and the sodium rushes in. The opening of these channels is triggered by reduction of the membrane potential, also known as depolarization. After the membrane becomes more depolarized and sodium ions have started entering the membrane, the potassium ions start leaving the membrane. A point now comes when the neurons have become depolarized to its peak, called action potential or spike potential. At this stage, the sodium channels become refractory, which means the channels become blocked and cannot open again until the membrane once more reaches the resting potential. Thus, no more sodium ions enter the membrane. After the action potentials or spike potential, the potassium channels are still open and these ions keep moving outside the membrane, thereby working to bring back the neuron at rest. They make the neuron membrane repolarized. Hyper-polarization. This is the final stage of action potential where the membrane potential overshoots its normal membrane potential of minus 70 millivolt. This overshoot occurs for a short span and comes to normal instantly. This overshoot occurs because of the excess outward movement of potassium ions, but this movement is soon balanced. Conduction of action potential. The movement of the message down the axon is called the conduction of the action potential. The action potential moves down the axon. As it travels, it remains constant in size. This phenomena can be understood in terms of all or none law. This law states that an action potential either occurs or does not occur, and once triggered, it is transmitted down the axon to its ends. An action potential always remains the same without growing or diminishing. When an action potential reaches a point where the axon branches but does not diminish in size. The reason why the size of the action potential remains the same is understood with the concept of nodes of Ranvier, which are the gaps between the myelinated axon. These nodes help in saltatory conduction of action potential. The axon does not carry one action potential throughout the axon, but it carries more than one action potentials, because the action potential generated at the beginning of the axons gets diminished at the end of one first myelinated segment. It needs to be generated again, and this generation of the action potential again is done at the nodes of Ranvier, which is of the same strength because of the same proportion of ion movements. This kind of conduction of action potential provides speed to the process of conduction of message, which is very essential for quick response. Because of this salutatory conduction, the message hops from one node to the other. It reaches faster than normal traveling at a higher speed. Communication between neurons. Main function of neurons is to transport the information. One neuron transports the information to another neuron and so on. This transportation of information is done through the release of neurotransmitter from one neuron and its uptake by another neuron. This process of transportation of messages from one neuron to another is called synaptic transmission. Synapses can occur in three places: on dendrites, on the soma, and on the other axons. The figure illustrates a synapse. The presynaptic membrane, located at the end of the terminal buttons, faces the postsynaptic membrane, located on the neuron that receives the message. These two membranes face each other across the synaptic cleft, usually a gap of 20 nanometer wide. The cytoplasm or the terminal buttons contain synaptic vesicles which carry the neurotransmitter. These synaptic vesicles are released by soma and are carried by fast exoplasmic transport to the terminal buttons. When the action potential is conducted down the axon, the terminal buttons release several synaptic vesicles located just inside the presynaptic membrane, fuse with the membrane and then break open, spilling their contents into the synaptic cleft. These neurotransmitters bind at binding sites of receiving neuron and change the membrane potential of the receiving neuron. Once binding occurs, the postsynaptic receptors open neurotransmitter dependent ion channels which permit the passage of ions into or out of the cell. Neurotransmitters open ion channels by at least two different methods, direct and indirect. In a direct method, a molecule of appropriate neurotransmitter attaches to the binding site and the ion channel opens up. The formal name for this combination receptor or ion channel is an inotropic receptor. These receptors, which are sensitive to the neurotransmitter called acetylcholine, contain sodium channels. When these channels are open, sodium ions enter the cell and depolarize the membrane. Indirect method is more complicated. Here, the receptors do not open the ion channels directly but start a chain of chemical events. These receptors are called metabotrophic receptors because they involve steps that require the cells expand metabolic energy. When a molecule of the neurotransmitter binds with the receptor activates a G protein situated inside the membrane next to the receptor. When activated, the G protein activates an enzyme that stimulates the production of a chemical called a second messenger. Molecules of the second messenger travel through the cytoplasm, attach themselves to nearby ion channels and cause them to open. Compared with postsynaptic potentials produced by inotropic receptors, those produced by metabotropic receptors take longer to begin and last longer. Postsynaptic potentials can be either depolarizing or hyperpolarizing, and it depends on the neurotransmitter which ion channels they open. The neurotransmitter dependent sodium channel is the most important source of excitatory postsynaptic potentials. When sodium channels are opened, sodium rushes in and causes a depolarization and excitatory postsynaptic potential. However, if potassium channels are open, some of the citations leave the cell, causing hyperpolarization and producing an inhibitory postsynaptic potential. Also, if the membrane potential has already been depolarizing by the activity of excitatory synapses located nearby, then The opening of chloride channels will permit chloride to enter the cell. This will bring the membrane potential back to its normal resting condition. Thus, the opening of chloride channels serves to neutralize EPSPS. These kind of postsynaptic potentials are brief, and they are kept brief by two mechanisms, reuptake and enzymatic deactivation. In reuptake, when an action potential arrives, the terminal button, it releases a small amount of neurotransmitter into the synaptic cleft and then take it back. In enzymatic deactivation, the enzyme of the neurotransmitter of the synaptic cleft breaks down the neurotransmitter and thereby deactivates it. Summary: neurons are the most important cells of the nervous system. They contain soma or cell body, dendrites, axon and terminal buttons. The internal structure of neuron contains lipids and proteins, cytoplasm, mitochondria which produce energy inside the neuron, and a deeply embedded nucleus which contains RNA and DNA. Supporting cells of the neurons are glial cells and Schwann cells. Glial cells are three types: astrocytes, oligodendrocytes and microglia cells. Schwann cells produce the myelin sheet of the neurons present in the peripheral nervous system. The communication within the neuron is an electrochemical process. It is accomplished in terms of the ion exchange. Important ions are sodium, potassium, chloride and anions. The membrane potential inside the cell membrane is more negative, -70 millivolt. The neuron gets stimulated when inside becomes more positive than outside. The communication between neurons takes place at synapse. The neurotransmitters from the terminal buttons are released and they are received by the receiving neuron at the binding sites, initiating a change in membrane potential. Excitatory and inhibitory neurotransmitters open different channels, and release of neurotransmitters in the synaptic cleft is very brief, and this is maintained by two processes, reuptake and enzymatic deactivation. Thank you.